Welcome to your very first AP Bio lecture. So I thought I would at least try to record as many of these notes as possible. I do feel that there's value in you reading the text and taking notes from it, but I also know that sometimes that can get a little tricky. So I thought I would create some notes for you. No video of myself, but at least you have my voice and you have the PowerPoint. So here we go. So here's a picture of a beach mouse, and we're going to use this beach mouse to learn a little bit about what biology can teach us. One of the things that we can learn is based on those four big ideas that we briefly talked about yesterday. So here they are. These are the four big ideas of biology, or of AP biology. If we break down all of biology into four things, the first idea is evolution. Now, evolution is the biggest idea and the biggest theme that we're going to see throughout everything, and we'll keep coming back to that, so that's why we start with that one. And then there's the second big idea of systems and homeostasis, so all of the different systems that plants, animals, any living organism has, and how do they maintain balance. And then big idea 3 is information and response. So not just nervous system information, but DNA and structural information, making proteins, making enzymes, and all of the ins and outs that are related to that. And then finally, interactions. And I guess you could use ecology in place of interactions. So how does everything work together? And again, we're going to sum it all up with evolution. Okay, this is a beach mouse, like on the first slide. And over here, we have an inland mouse. They are of the same species. They're both Pyromycus pollinitis. And you can see, let me pan back out there for you, that both of these mice look similar except for their colorings. Well, this beach mouse, he lives on the beach where the light coloring helps it to blend into its surroundings. Whereas the inland mouse lives in areas where there's thick vegetation and soil, and the dark coloring is going to help that mouse to blend into its surroundings. So when we think about biology, we can talk about how these mice were able to have two different colors, how that came about based on where they live. So we're going to start with the idea of there being 5 unifying themes of biology. Now, every textbook that you look at, they're going to have different themes, different ideas, but it's the same basic principles. So I've seen up to 7, I've seen as little as 4, but we've got it divided into 5 basic themes, and those are organization, and along with organization and new levels of organization will come new properties. There's expression and transmission of genetic information, i.e. DNA, and then there's the transfer and transformation of energy and matter, so if we're thinking plants, photosynthesis, interactions of organisms and their environments, and then again that big idea of evolution. So here's with the first one. So first we have the different levels of organization. We start with the smallest being atoms, working our way up to molecules, then to cells, tissues, organs, organ systems or body systems, or an organism, the population, which is a group of similar species or organisms, and then the community, which includes all of the organisms, and then the ecosystem, where we're adding the non-living parts of the environment to, finally, to the earth or our biosphere. So when we take a look at all of those systems and all of those different levels of organization, we can apply basically 2 different ideas to look at that. The first idea is going to be called reductionism, And that means that we take a complex system and reduce it to more manageable components. So we're taking the ecosystem and we're only looking at a part of the ecosystem, so the living part of the ecosystem. And then we're getting smaller and smaller, and we're looking at just a certain population. And then maybe we're only looking at certain individuals. So we just keep getting smaller and smaller and smaller as we ask and answer more questions. A great example of this was Watson and Crick when they studied the DNA. So they wanted to know if the structure of DNA had anything to do with its properties and how we were able to inherit our traits. So by discovering the structure, they were able to apply reductionism and give us a better understanding of our heredity. Now, this doesn't always work, so sometimes we apply the process that's called emergent properties. And this is just the opposite of reductionism, where reductionism was going from large and smaller, smaller, smaller. Now we're looking at what is coming out of each new level. So every time we move up a level, from atoms to molecules, from molecules to cells, from cells to tissues, we're gaining properties that we can study. So every new level has new and different, that's novel, properties that were absent from the previous level. Tissues can work together to do things that an individual cell cannot do. I know some of you don't like plants, but a great example of this is in plants and photosynthesis. It happens in a chloroplast, which is a small organelle inside of a plant cell. The chloroplast has inside of it chlorophyll and some other molecules. If you put chlorophyll and those molecules in a test tube, it doesn't work. We don't get photosynthesis. But if we are to take all of that and make it into a chloroplast, then we get photosynthesis. So that would be an emergent property when we combine those things together. I have a picture of a bicycle here because I wanted to give you an example to remind yourself that a bicycle is made up of many small parts. You've got the tires, you've got the pedals, you've got the handlebars, and independently, each one has its own properties. But when we put it all together, a whole new emergent property arises, and that is the ability to move us forward. Of course, it takes us and some work. So what we end up employing most of the time, instead of just a simple reductionism or emergent properties, is a combination of both. And we combine that to call it systems biology. So we explore a system by analyzing all of the interactions of its parts. And that can hopefully lead us to new and more open-ended questions. So some examples of that: how does blood pressure medicine affect other organs? So sure, fine, you've got this medicine that's going to lower your blood pressure, but what does that do to your liver? What does it do to your kidneys? What does it do to you as a human? Okay, so we have to look at a whole system biology. We don't want to just treat one thing and then end up having lots of other problems because of that. And the other thing is that we can look at, another example would be higher CO2 levels in an ecosystem. How does that affect both the living and non-living components of the ecosystem? When we look at systems biology, and especially emergent properties, what we end up discovering is that there is a correlation between an organism's structures and the functions that an organism can do. One example of that are leaves. Plants' leaves are very thin and they're flat, which allows them to be able to get the most sunlight absorption or light absorption possible. Another example is the hummingbird. The hummingbird actually has shoulder joints that can rotate, okay, which allows them to move their wings backwards, whereas other birds can't quite do that. They can move forward, but they can't move backward. This allows the hummingbird to be able to hover in the air and to be able to get into the parts of the flower. We can also look at how its mouth parts are designed specifically for certain flowers. And then finally, with the levels of organization, the last thing that we should talk about is the cell. You've all had basic biology and have gone over all the different organelles and their functions and all of that. So we won't go through that. If you need to review it, you might want to just spend a few minutes, reminding yourself what the nucleus does, what the mitochondria do. But basically, the cell is an organism's basic and smallest unit that can perform all life functions. So now there still is some debate on whether viruses would be considered living. Viruses don't have cells, so therefore we can't consider them living. There is one thing that all cells have in common, and that's a membrane. Some cells, like bacteria cells, plant cells, have an additional layer of protection, which is called the cell wall, but all cells do have a cell membrane. There are two main types of cells, the bacteria or prokaryotic cells, which have no nucleus and no membrane-wrapped organelles. And then the eukaryotic cells, which you have. So you have eukaryotic cells, and they have a nucleus and they also have membrane-bound organelles. The second of our unifying themes in biology is information. We know that our information inherited is contained in DNA. Well, DNA is formed by wrapping around proteins into structures called chromosomes, and it's the chromosomes that we are inheriting. When the sperm and the egg combine, 23 sperm chromosomes, 23 egg chromosomes in humans gives a full set of 46. Every time a cell needs to divide, it's important that DNA is replicated or copied exactly so that each new cell has the same exact information as the parent cell that gave rise to it. On each chromosome, we can take a look. I'm going to zoom in here. We can take a look at each chromosome, and if we uncoil that, we'll get little sections that are called genes. Okay, so when we hear like it's in your genes, so that's just a little section of DNA that's red, and then we've got the whole DNA. Okay, so a gene is a little section of DNA that codes for specific information that's going to be transmitted to that organism. So let's take a look at an example from our alphabet. Now we have 26 letters and DNA only has 4, but I just want to give you an example of how this can happen. If we take the letters R, A, and T and we put them one way, we get the word rat. Ew. If we put them in a different order, we get tar. And finally, we can arrange them in yet a third order to get art. So DNA is very similar to that. We're working with adenine, guanine, thymine, and cytosine, so 4 different letters or bases, and we can arrange them in different ways to produce different proteins, to produce different enzymes, all of the things that our bodies need. So it's kind of like the alphabet making words. And we have, how do we get from that alphabet to what we look like? And that's called gene expression. So gene expression is when the cell takes the information that's inside that gene, that A, T, G, C, in whatever combination, and it uses it to make certain things. Now, when we see similarities throughout all different species, we know that there is a certain sequence of nucleotides, like A, A, G, G, C, T, C, that would code for the same thing in one organism to the next, whether you're a mouse, a jellyfish, or a plant. There is something that creates a cell membrane. Okay? So then we can say that the differences in organisms reflect differences in their sequences, not DNA. So remember again, DNA, A, T, G, and C, same 4 letters, every living thing on this planet, but the differences in you and a jellyfish are going to come from the differences in your nucleotide sequences. So I put a picture of some cars down here just to kind of remind us or give us a point of reference that just like a car is a basic structure that gets you from point A to point B, there are different ways that those cars are put together, different colors, different size engines, all of that good stuff that makes them all slightly different. So a Corvette is definitely different from this little Hyundai, whatever it is. We can study something called genomics, and this has become very prevalent in the last few years as we've uncoded or decoded very many different organisms, the human genome being completely discovered. It's where we have our entire set of instructions, and we use computers to determine what those instructions are. Literally pages and pages of A, T, G, C, just over and over and over again. And so for us, we have about 3 billion pairs of nucleotides. So yes, 6 billion nucleotides from each parent. That's pretty easy math to do, but just to kind of give you that number, 12 billion nucleotides would be what our genome is made of, our entire set of instructions. When we study genomics, we're studying all of that information at once, and sometimes in more than one organism, and we can call that bioinformatics when we're looking at an ape, a mouse, and a giraffe together at the same time. We could be studying bioinformatics. Okay, our third theme out of 5 is energy and matter. So the biggest point of energy and matter is that everything an organism does is work. It doesn't matter if you're raising your hand, if you're sitting down, if you're eating something, it all is work, and it all requires energy. We get our energy from the food that we eat. Plants make their energy from taking in sunlight. The actual input of all energy is the sun. Okay? The sun inputs the energy into the plants. The plants convert that energy, or they transform it into something that's usable, or for us, eatable. Okay? As they're doing that, they're actually releasing heat. The organisms down here that eat those plants are taking the energy from the plants and they're transforming it into another form, so into muscle or fat or energy. Sorry about that brief interruption. And then that organism also releases heat as it's burning its energy or using its energy to run, to sleep, to do other activities. Those organisms can be eaten by other organisms, which will convert the energy into something they can use, the energy in the muscle, and they'll release heat. And then finally we have the decomposers, like the bacteria and fungus, which will go to work on a dead organism and convert that matter into a different kind of energy for themselves, also releasing heat. So it's a big cycle of taking in energy, transforming it into something usable, and then releasing heat as part of the process. Okay. Our fourth theme of biology are interactions. This one's pretty straightforward. Each organism we know is continuously interacting with other living things in its environment, and the non-living parts of its environment too. When you take a drink of water, you're interacting with a non-living part of your environment. Some interactions between species can be mutually beneficial. Okay, both organisms get a benefit from it. A sea turtle can have sturgeon fish that will eat all of the little parasites off the turtle, so the turtle gets to be parasite free, and the surgeonfish gets to have a free meal, and then also get a little bit of protection. Okay, so that would be an example of mutually beneficial. And then there are other ones that cause harm, like parasitic, when we have a tapeworm and that causes us to be very sick. Or some other organisms can get cysts in their brains, and that can actually kill them. Well, if it kills the host, it also is killing the organism. So in that way, it's harmful to both of them. So here's just a little picture of some interactions and of a basic food chain, and how you can see that there's always things going on. So we have producers up here, the plants that are using the sunlight to make food, so there's an interaction, and they're taking in water, and they're also taking in carbon dioxide. The fish is going to eat the producer, so it's interacting that way. It's also releasing chemicals into the water from its excrement. Okay, so that's another way it's interacting. Those chemicals are settling down into the sediment, which is another interaction, so on and so forth. Okay, that brings us to our final theme of evolution. And again, we said that was going to be our biggest theme. It is the core theme, and we can use evolution to explain why things seem to look the same, but also we have so much difference and diversity. As you can see in this picture, we've got the skeletons overlaid over what the organism actually looks like. And you can see all of these organisms have a skeleton, and it looks very similar in all of them. They all have tails, and they all have skulls. Okay, so we've got that unity, but we've also got diversity. We can also use this to explain why organisms seem to be perfectly suited for the environments that they live in. So let's take a little bit of a closer look at evolution. There are 3 domains to life. We call those the archaea, which are the extremophiles. They live in crazy, weird environments. The bacteria, which we encounter most. Okay, they can be harmful or helpful to us. And the eukarya, which are all of the other kingdoms of life. So the plants, the fungus, the protists, the animals. Now, we don't really have a consensus on exactly how all of the organisms should be grouped together, except that we do have a scientific consensus on there are 3 domains that are very distinct from one another. So we've done a lot of updating and a lot of comparisons of DNA in the last couple of years, which has caused us to possibly rethink that 6-kingdom model and go with a more broad 13 to 15 kingdom model. So what we're going to ask you to understand is that there's 3 domains, the archaea, the bacteria, and the eukarya. So let's talk about Darwin a little bit and his theory. In 1859, the world changed when Darwin published his book on the origin of species by means of natural selection, usually just referred to as Origin of Species. In Darwin's book, he had 2 main points. The first was that there seemed to be descent with modification, which meant that one species had ancestors that were slightly different from them. So you are not exactly the same as your parents, who are not exactly the same as your grandparents. So there is descent with some modifications. Okay? And then the second main point that in his book that he makes is natural selection, which is what he called the mechanism for this descent with modification to happen. So this helped descent to happen. The things that were good and useful in the environment were selected for because they help the organism survive. Darwin developed his theory from many, many detailed observations over many years. Okay, so the first thing before I get into Darwin's observations, I just want to apologize for the grid-like design on this. When I made the PowerPoint on my computer, it didn't look like this, and then transferred it to the iPad. So I realized that it might be a little bit of an issue to read through the lines, but I won't do that again. So I won't use this template again. But so let's talk about Darwin. So Darwin made 3 observations. The first was that individuals and populations can vary, and we know this. We can look around the room and see that we have a population of students and they're all slightly different. And if we were to look at their parents, they seem to get their traits from their parents, so those are inherited. Darwin also noticed, and we can too, that a population is able to produce more offspring than can survive to that reproduction age. Frogs lay hundreds of eggs, and only a small percentage of those will actually make it into adulthood to make more baby frogs. And then he also noticed that species seemed particularly suited to the environments that they lived in. And these 3 observations caused him to wonder. From those 3 observations, he made something that's called an inference. So he inferred, or looked at and made a really good, justified reason for why this happened. And he said individuals that have inherited traits that are more suited to their environment are going to survive because they're suited to their environment. They're going to survive to the age to be able to reproduce and find a mate. Okay? And if they are able to find a mate and reproduce, they are able to pass on their traits. And over time, those traits are going to become more numerous in the population. All summed up with 2 words, natural selection. Here is an example of some finches and the tree of life. So this is kind of an example of a cladogram or an evolutionary history or a branching pattern that's going to show how this one organism may have given rise to all of these organisms. So it all started with one, and then small changes, mutations, were made that caused each one of these to be best suited to its environment. And we have our last section for Chapter 1, and that's biological inquiry. At the root of the word science is the idea of to know, nous, to know something. When we do science, like we're supposed to, we're performing an inquiry or a search for information and explanations to the things that we see. A lot of the labs that we do in AP, we're going to do a lab to get you an idea of what to do, and then you're going to have what's called an inquiry piece where you're going to get to decide the variables and the factors that you choose and a question that you want answered. In middle school, you may have studied the quote-unquote scientific method, and there really is no set forth laid plan for exactly how that has to happen, but a good inquiry in science includes several of these things, or maybe even all of them. There have to be some observations that are made. You have to form a hypothesis that makes sense, so a guess as to what will happen that is logical and makes sense. You have to be able to test your hypothesis. You can't just come up with an idea, and if you can't test it, you have no way to say whether that was proved, not proved. We don't really want to use those words, so accepted, rejected, okay? We need to be able to collect some data, so that can be either quantitative, as in numbers, or qualitative, as in things that we're observing about behaviors or anything related to that. We should be able to use some inductive reasoning, where we're going from specifics to generalizations. So if we do a study, we should be able to look at the specifics of that study and make broader generalizations about a population. But we should also be able to use some deductive reasoning where we take generalizations and then break that down into more specifics to find out what's going on. When we perform a controlled experiment, sometimes this gets confused, thinking that you have to quote-unquote control every little piece of everything, and that's not true. So a controlled experiment is just an experiment that compares one group, which is usually your experimental group, to another group that you're going to call the control. Usually those two groups are only going to differ by one factor or one variable. The more variables you get mixed into it, the harder it is to actually make a correlation between the two things, okay, or three things. What usually people are talking about when they say controls, they should be referring to things called constants. These are things that the experimenter is going to try their best to keep the same from one group to the next. You don't want to test a hair growth serum on 40-year-old men and have your other group be 20-year-old men, because there are going to be some differences between those two. So trying to hold things constant. You're not going to want to grow one group of plants in bright sunlight and the other in not so bright sunlight. So holding things constant as much as you can, okay? But you know what? In field research, when you're out in nature, that's not always possible. You have no control over nature, and sometimes you're getting a lot of qualitative data when you're out there. And then we need to talk about what is a theory. Theory is thrown around a lot. I have a theory about why so many kids are in the hallway. I have a theory about why my mom does this. That is an untested speculation. I think this, okay? That's not what a scientific theory is. A scientific theory is bigger than a hypothesis, and it's usually supported by a wide body of evidence, lots of different pieces or lots of different hypotheses that have been accepted go into making a theory. And sometimes a theory can change. In science, truth is conditional until we find something that disproves it. Science has its limitations, though. One thing about doing a scientific inquiry is that a hypothesis must be falsifiable. You must be able to say that this is not true. So for instance, if you have a flashlight that doesn't work, you go to turn on your flashlight, you're out camping and it doesn't work. You could make a hypothesis that it's the batteries that are causing your flashlight to not work. Well, that hypothesis is falsifiable. If you take the old batteries out and put brand new batteries in, and your flashlight still doesn't work, your hypothesis was false. It was not the batteries that were causing your flashlight to not work. Okay, so we have to have a falsifiable hypothesis. And another thing with science is we can only use natural explanations for natural phenomena. We're going to talk about evolution a lot in this class. There is always going to be some contention over whether evolution is true versus other theories. But what we're not going to do is we're not going to look at supernatural and religious matters. They are not falsifiable. Okay, so we're not going to look at that. Those are issues of faith, and that goes outside the bounds of what science can do. So if you believe that, in our flashlight example, that there was a ghost that was causing your flashlight to not work, there is no way that we can prove that it wasn't that supernatural being, that ghost. So therefore, we're going to look at things that are within nature, natural explanations for the things we see. Hopefully that wasn't too terrible, and again, I'm sorry about the grids. I will make sure that the next PowerPoint doesn't look like that. please let me know if you have any further questions or any suggestions for how you think this went, what you want me to do more of, less of, etc., etc. Talk to you in class.